Hello everyone and welcome to the next lecture for MZHE Biology Preparation Series. We are still in section 1 and today we are gonna go to the second last section, which is how you would go for the different microscopes and what to do and the basic parts. It's an easy unit, it's a very scoring unit, so make sure you know it. Don't skip any question based on it. So let's quickly start with this. So the first thing you have to know in this is the basic parts of a light microscope. Okay, you must be knowing for your syllabus or for your course, you have to know that there are two basic types of microscope, the light microscope and electron microscope. So we've started with the light microscope first before we go into the differences later on. So here when you this thing, the big long thing, it's called an eyepiece tube, and you have a lens here. It's the eyepiece or the ocular lens. Please remember, lenses are always there for magnification. So you have an eyepiece lens and you have an objective lenses here. So you have lenses in two parts in the light microscope. Okay, so this is the nose piece, which is not very important, but you should know the basic part. This is the arm. Okay, this is the stage where you put your slides, you clip them with the stage clip so that the slides doesn't move when you are observing the specimen. Just below the slide you have something called the diaphragm. The diaphragm is something which controls the amount of light entering your specimen. And where does the light come from? It comes from something called an illuminator or a light source. After that you have two knobs here. Different light microscope can have different settings, but these are the basic settings. They might give you some other diagram in the exam. It doesn't matter, they will have the same basic parts. Okay, so the small knob that you see here is the fine focus knob or the fine adjustment knob, and the big knob that you see here is the coarse focus or the coarse adjustment knob. So what is this one for? This one is just focusing the sample. Okay, once you see the sample, you start focusing it finally. This is for the fine details of the sample. So this is just for bringing your sample into focus, and this one, when you're tuning it, you're getting a much clearer and detailed picture of it. Okay, again, this diagram talks about what each one of the adjustment knobs do, or what is the purpose of the ocular lens. It's just what I have explained. It's the same concept. Okay, the same thing. We've already discussed the major portions of the light microscope. Quickly going into electron microscope. Now electron microscope is of two types: scanning electron microscope and transmission electron microscope. These are the types which will be asked in the exam. So SEM is actually for giving you the details only about the surface of cells. You will not get any information regarding the internal structures, but TEM is giving you all the details of the internal structures. Okay, the magnification is less for SEM and it's too much for TEM. You can see it's like 50,000x here, and it's like up to 2 million times, which is very high. Okay, and then scanning electron microscopes gives you a 3D image, but the transmission electron microscopes gives you a 2D image. Okay, coming to the differences now between the light microscope and electron microscope. The basic difference first is this uses light, this uses electrons. Okay, and light microscope is relatively inexpensive. It doesn't need more training. Electron microscope are expensive, they're large, you cannot carry them from one place to the other easily. It requires training to use them. But what is more important here to know is in light microscope you can see color images, whereas in electron microscope you can just see black and white images. In light microscope you can see living specimens, where in an electron microscope the specimen has to be dead. Okay, and coming to which one is better in terms of resolving power or magnification, we will see the electron microscope is much better. Okay, next you have to know about the magnification of a microscope. Magnification is basically how big would the object be, okay, or your specimen be, how big you can make it to see. So magnification is calculated by multiplying the power of the objective lens with the power of the eyepiece lens. For example, if they say the power of the objective lens is 4x and the power of the eyepiece is 10x, then what is the total magnification? You just simply multiply it to get your total magnification, which in this case is 40x. And let's look at some of the questions. So here they say biology teacher has assigned you to study the internal activities of the cell's mitochondria. Remember, they are talking about the internal details, not the surface details, okay? They said you have different microscopes in the lab. Which one will you go for? So we know whenever we have to go deep into the cell to see the internal structures, we cannot use a light microscope. Light and compound microscope are basically the same, okay? Scanning will only give us details about the external surface area, and transmission is the one which we use for looking into the internal details of the cells. So here the answer would be transmission electron microscope. Coming to the next question, it's a long question, but just look at the important parts here. They say the magnification this time is 1000x. They did not, and they said that first of all the student examined the leaf cell under an electron microscope of this much magnification, which is 1000x, and they said they tried to examine the same leaf, now this time under a light microscope, which has a magnification of 10x ocular lens and 100x objective lens. So if you see the magnification of electron microscope is given, which is 1000x, you have to calculate the magnification of light microscope. You just multiply these two terms. Now we see even the light microscope has a magnification of 1000x. Now what? Now they said, which of the following statements supports the student conclusion? Let's look at it. The resolution of the two microscopes is the same, but magnification used, electron microscope given image 10 times larger. No, we just found out the magnification is the same. So this is not what are we looking for. The student used the electron microscope at a higher magnification. Again, no. The electron microscope has less resolution, but the use of electron will help to obtain. So we always also know that electron microscope will always have a high magnification and a high resolution power compared to the light microscope. So it can never have a less resolving power or less resolution at all. And now let's see, the magnification used in the two microscope is the same. Yes, 1000x, but the electron microscope has better resolution than the light microscope. This is the only sentence which makes sense. So your answer would be answer number D. Okay, option number D. The next question is, transmission electron microscope is the best available microscope because it gives what, 3D images? No, it's the SEM, the scanning electron microscope. Detailed images of the cell structure? Yes, we know it gives us a detailed image. Anyways, let's check out the other ones. Uses dyes to produce colorful images. We know light microscope do that, right? And uses light and can be used anywhere. Again, it's a light microscope. Okay, so light microscope, we can see, as I said, we can see colorful living specimens. So the only answer which is possible here is option number B. Coming to the last question, here they have given you a picture of a light microscope or a compound microscope. And let's look at the basic parts. A is of course the ocular lens. The B is the eye tube. Then C is a part of the arm. Okay. D is the stage, and I think this is the condenser or the diaphragm. Okay, now they're asking you to focus a specimen under high power. Which label part of the microscope would you most likely be adjusted? Now look here, this one eyepiece has nothing to do with observing anything with high power. It's something to do with something which has a lens. C again is part of the arm, nothing to do with resolving power magnification. D controls the amount of light, nothing to do with high power thing. The only thing which has lens in the whole structure is option number A. Okay, the part A. So your answer would be A. With this, we finish off this lecture. In the next lecture, we'll be talking about the different types of cells, cell organelles, and we will also be talking about the cell transport and try to solve some MSAT questions. Thank you so much.